So today I'd like to mention a bit one of the applications that we mainly or mostly used in our laboratory. I think we are using this server maybe more than 4 or 5 years. And today I'd like to just give you a brief theoretical background about this application, which is called Metacore Metadrug. And then I'd like to give 1 or 2 examples that we use this this server, which is available, and how we used this server in our drug design applications. So during the talk, if you have any questions, please stop me and we can discuss further. Server is called Metacore Metadrug, which is a binary QASR model. And I would like to just mention what it is and how it is actually working. So Metacore Metadrug, as I said, is a server that uses the structures of the small molecules and their corresponding physicochemical properties. So at the first workshop, we have listened QASR, which is the structure-activity relationship, and we are using mathematical correlation, and we are building a mathematical model to represent the structure-activity relationship in a quantitative way. And this one, in the Metacore Metadrug, we have about 70 different QASR. So when I say QASR, actually it's the QSPR, which is the quantitative structure property relationship. So this means that we can create or we can build a model or ligand-based model based on any physical-chemical property, such as if you use, let's say, if you have enough data with structure with the cardiotoxicity, in that case, you can create a toxicity QASR model. If you have lipophilicity, for example, and if you can measure the log p values, in that case, you can create a QSPR using these models. In short, actually, we are mentioning these QASR models, but as I said, it is not the real activity, but it is related with the property. So as I mentioned here, that we used in the Metacore, we have in total around or more than 70 different QASR models, and I will explain how this model is actually created. So here you can find one simple example actually that shows you how the QASR models or QASR table should be. So here we have a structure, right? The structure can be, in the QASR, it can be 2D QASR as we are actually using. So this means that we have just 2-dimensional structures. So it can be represented with the SMILE codes or STF codes, or let's say just the representation of the molecules, but their bonded representation. Then we need to get the descriptors, right? We need to use the molecule descriptors, and then we need to get this QASR table. So here in the first column, you will find the compound names or the structures, and then their activities. Here, activities, as you can see, represent with the 1 and 0. So that's why we call binary QASR. So as Selma already mentioned, here we are using the binary code that if we have the unit of the structures that is on the used descriptor set, then we are saying 1, and if it is absent, we are using 0. We are passing to a bias theorem, a kind of probability analysis, and we are getting the binary QASR models. And of course, when we get this, we need to use an extensive set of statistical methods to validate our models. And I will show you what kind of statistical analysis method that we are using to validate the models. So it is quite simple to use, I can say. So what you need to do, you need to go to the server and just upload your structure, or you can draw, by the way. I think I can show you in a simple way. So yeah, if you just type to portal.genego.com, in that case, here you can find upload the structure, right? You can upload the structure, or as I said, if there is no structure available in your case, you can also draw here. So let's say, let me just draw a structure here. So quite easy to draw. Maybe I can put it here, a carbonyl group. And then maybe here I can put one OH. So if you draw your structure, here you can see metabolite generation options. We will see in our presentation with Metacore, you can actually get the first pass and the second pass metabolites. So the metabolites is quite important issue in the drug design. So let's say if you found a small molecule that is fitting very well to the binding pocket, very active, and you have the very low energy as a delta G, but if it is metabolizes at the liver, then everything should be reconsidered from beginning. So here we need to get the first pass and second pass metabolites. So for these molecules, for example, we will search the first pass and second metabolites. So in the first pass metabolism, mainly oxidation and reduction reactions will be considered, but in the second pass mechanism, mainly the transfer of the glucuronide and yeah, you can. So different type of enzymatic reactions can be considered. So this is the part. So we decide, we just draw our structure or we uploaded our structure. It can be STF or MOL2, by the way, the file type. And you don't need to have only one molecule. So let's say if you have 500 molecules, then you can upload all these 500 molecules. I mean, you need to, of course, merge all these 500 molecules to one single file. So you need to merge them. And then you can upload this data to here. And then you can get all of the information together. So here, let's say if you have more than one structure, because at the end we will get a one report. So if you click this, you will get only one single report for all of the STFs. But if you don't click this, in that case you will get all reports individually. So next. So it will understand the structure. And then here we have similarity search. You can actually increase or decrease the number from 0 to 1. So here our default number is 0.7. So this means that it will be searched the similarity between your compound that we gave and the FDA approved compounds in the system. Here, we can also select the properties to calculate, such as molecular formula, molecular weight, Lipinski's rule of 5, hydrogen bond acceptor, donor groups, reactive groups. And then here, if you want, you can also go with the CIP 450 QASR models. Here, there are different QASR models that these are related with the metabolite predictions. Here you will see different QASR models again, such as hERG binder. The hERG blockage is quite an important issue in the drug design. So let's assume that you identified very active molecules, but if this molecules goes and binds to the hERG channel and blocks the hERG channel, in that case, hERG channel is a potassium ion channel, and blockage of the hERG ion channel leads to decrease of the ions, potassium ions passing from the intracellular cavity to extracellular cavity. So this actually leads to the long QT syndrome, LQTS. And finally, it's actually leading to the sudden death or, let's say, cardiotoxicity. So for that, it is quite important to check if your molecules will have high affinity to the hERG channel. So you can do with different ways. So currently, hERG hERG protein structure is available. I think in 2017, PDB structure becomes available, so you can do docking instead going to check the QASR. But you can also, without making the docking, you can get an idea about the activity of your molecules against the hERG channel. So here, values actually giving in this range, minus 1.7 to 5, 0.5 to 1, and so on. Sometimes you can see the values normalized between the 0 and 1. So let's say if it is activity, therapeutic activity, in that case, higher than the 0.5 means we may have some potential against this disease. So let's say if you have cancer model, in that case, if your molecule receive, let's say 0.7, so this means that around 70% of probability that your molecules will likely to be an anti-cancer compound. But if it is cardiotoxicity, of course, in that case, we want lower number. So in that case, it will be, if it is, let's say, less than 0.5, in that case, this means less toxic. Sometimes you see this kind of numbers, 1.7 and 5. So these numbers are based on the IC50 values or KD values. Usually, if you have 50 micromolar of IC50 and higher, so let's say 50 macromolar, 100 macromolar or more, so in that case, this means that more or less your molecule is not active. So it's very weak binder. So if you get the log 50 micromolar, you will get about 1.7. So these are the numbers that actually we want the range between that. So if your molecules between the 0.5 and 1, that's okay. But if it is outside of this range, this means your molecule is not actually fitting well to these criteria. Then, yes.
Yes. Why it is minus and why the number is negative?
Here.
Yes.
Yeah, this is for the specific aims. I mean, if for each of the models, if you go here, actually it is giving you the description of the model. So this is the human soluble epoxide hydrolyze inhibition PIC50. Cutoff is minus 1.7. So this is the cutoff that it is actually received. But you can change it, right? So you can, based on your values. I mean, if you want higher cutoff, higher barrier, you can actually use it. Sometimes in our cases, for example, although the cutoff value of therapeutic activity value is 0.5, we are using maybe 0.75 or 0.80 in order to become more sure about the activity of the molecules. And again, for each molecule, you can see here the statistical results, statistical data about the model. So, for example, this model is constructed with 675 compounds. And its R-square value is 0.92, which is quite high, and its root mean square error is about 0.5. So you can also find this kind of statistical results for each of the model. Yeah, the second thing that I'd like to mention is the ADME. So here you can also get idea about the blood-brain barrier. Again, if you are specifically working on the central nervous system diseases, in that case, sometimes you want to enter the molecule to this central CNS, and sometimes you don't want. So based on your research and based on your project, you need to check if your molecule is actually passing the blood-brain barrier or not. So here, again, we have two different values. So this means that if it is between minus 0.3 and 1.5, this means that your molecule can pass the blood-brain barrier. But if it is, let's say, higher than 0.3 and more than 1.5, then if it is outside of this range, in short, then it is not passing the blood-brain barrier. Again, this one is also quite important. So if it is passing, this is also quite important. Affinity to human serum albumin log value. This is the retention time. So positive values corresponds to higher protein binding. So sometimes, when you give the molecule orally, it goes and binds to off-target molecules. So one of them is the human serum albumin. So it is very well-known protein that the molecules can go and bind to this protein. In that case, in order to reach the enough amount of concentration to your own target, you need to maybe use more concentration, more dose. So for this, you need to check if your molecule will be able to bind specifically to your own target, or it may go and bind to this kind of off targets. Okay, so this actually gives you an idea, and also, okay, it may bind to serum albumin, human serum albumin with a high affinity, but if it is stay short time, if the retention time is short, then it may not create a problem. But sometimes it may bind to human serum albumin with a low or weak affinity, but if the retention time is high, again, this may create some problems. So you need to check this as well. So this model actually gives you about the retention time and the binding affinity. So here it is the retention time, how long your molecule stays in the human serum albumin, and this one actually tells you what is the potential of your molecule staying in the binding or binding to the human serum albumin, and this is the percentage. So if it is more than, let's say, more than 50%, so this means that your molecules more likely will bind to human serum albumin. Again, water solubility and the lipophilicity of your molecules are quite important. In our we have academic work here, but also I have my own company, and within my own company, we are also doing these drug development studies, and the one of the most difficult part that we are actually facing is the water solubility of your small molecules. So in in the biological environment, or let's say in vitro environment, you are using DMSO or maybe high log p-value solvents, or maybe ethanol and so on. But in real life, of course, if you are taking the drug with orally, it is better to know if your molecules will be soluble in the water or not. So let's say that your molecule is very lipophilic and it is not dissolving well in the water environment. So this will create a lot of problem, right? So you need to find a way to increase the solubility, water solubility. Yeah, I can ask here a question to audience. What are the ways to increase the water solubility of your molecules? Any idea? Well, if you make them polar, then you are actually changing the molecule. You don't want to make the.
We can solve.
Salt. Okay, is this possible for always? Making it salt?
Not for all that.
If not, what you will do? If there is no any place that you can change if the pHs are not suitable to make the salt? Carrier. If you put a carrier, can you give it the oral way? No. Sometimes. If you add aliphatic groups, then you are changing the structure.
Just delivery systems.
You can use the delivery system, but you want to give the IV or PO?
Using buffer. Buffer.
Okay. If you use buffer, will this help to increase the solubility, or how you will make the how you will formalize the molecule? Okay, considerable. Yes? If you use nanoparticles, can you give the molecule as an oral way? Comfortable is quite important, right? So taking the drug, taking the pill in the home is different. Going to the hospital, getting the injection is different. So lots of companies are now considering oral drugs. This is the reason. I mean, you know, you can make lots of solutions with the IV, but what we want oral drugs. Go to home, take you know, a glass of water and take your pill. That's all. But if it is IV, then everything becomes uncomfortable and difficult. So this is the case. So that's why still, because although there are lots of advancement in the biological drugs, still lots of companies actually investing money to oral drugs, small molecule drugs. So for that, we need to know if our molecules actually water soluble or not, right? So here again, this model actually gives us water solubility at 25 degrees of Celsius as a milligram per liter, and cutoffs are from 2 to 4. And if your molecules actually sit in this number, between this number, then maybe you can be estimate that your molecules will be easily soluble. Yeah, maybe this is the most important part, prediction of therapeutic activity. So here we have about more than 25 different diseases, and like depression, bacterial, cancer, HIV, heart failure, obesity, allergy, Alzheimer, Parkinson. It goes like this, and so on. And you know, as I said, if you give one molecule at the beginning, and if you want to predict all of these diseases, then you should just click here, and then at the end you will get information about this. Okay, so this is the case. So we can go and check each of them, such as, if you want, we can go cancer. So this is potential activity against the cancer. Cutoff is 0.5. Values higher than 0.5 indicate potential active compounds. Training set consists of approved drugs. Model description. Training set is 886. Test set is 167. Sensitivity is 0.89, specificity is 0.83, accuracy is 0.86, and multiple coefficient correlation is 0.72. So very good, actually. I mean, I can say that.
How about the target?
Hmm?
Do we have a target?
Yeah, I will mention this. Yeah, I will mention this. Okay, so as I said, it is the values are normalized between the 0 and 1. Sometimes you will get 0.2, 0.3, sometimes you will get 0.8. So it will give you some idea. Another thing is toxic effects. So here again, we have around 25 different toxicity, such as AIMS, cardiotoxicity, genotoxicity, hepatotoxicity, nephrotoxicity, neurotoxicity, liver cholestasis, liver necrosis, liver weight gain, kidney problems, testicular toxicity, pulmonary toxicity, and so on. So there are a lot of toxicity that you can have. As I said, the values should be between 0 to 0.5. Sometimes, I think in a few of them, we have different cutoffs. So each of them actually giving explanation. So here, growth inhibition of the MCF7 cell line, human breast adenocarcinoma, PGI-50, cutoff is 6, values from 6 to 8 correspond to a toxic metabolite, and values less than 6 are preferable, values less than 3 are more favorable, and so on. So one can actually get this information. Anyway, let's go to here, prediction of therapeutic activity, and maybe should we go with the toxic effect? No, let's put this. So if you go with the next. Yeah, it started. That's all, as you can see. So the job is started, and then I think it will take about one or 2 minutes, and then I will show you the result. But now let's go and check the theory. Yes, we mentioned this. So the goal of the Metadrug is to convert a vast amount of available data into a comprehensive knowledge about the chemicals using chemoinformatics and system biology tools. It includes similarity search, metabolite prediction, physical chemical properties calculation, QASR models for therapeutic activity and toxicity, extensive database of human ligand-protein interactions, ADME. And in order to generate a Metadrug report, a typical Metadrug workflow requires a SDF file or MOL file, as I showed you. And it is actually creating lots of QASR models. So as I described, metabolite prediction is quite important. So when you get the oral drug, right, it is goes to GI system, and then it actually goes to portal vein to liver, and then from liver to systemic circulation. So when we go to here to the liver, then the enzymes at the liver may lead to the metabolism. So this is actually quite important problem. So if you start with a small molecule, then this small molecule maybe goes to breakdown of the molecules, or some subgroups may be added because of this metabolites. So sometimes we can have the first pass metabolism, and if after getting the blood circulation, if the drug can be metabolized in other tissues and reach the liver second time, the liver can metabolize the molecule second time, and this is called second pass metabolism. So in the Metacore Metadrug, we can predict both the first pass metabolism and the second pass metabolism metabolites. So in the first pass metabolites, it is using the oxidation reduction hydrolysis using the CYP450 enzymes, and in the second pass metabolism, it includes conjugation reactions. Yes, the rules were actually how we are calculating this. The rules were derived from the analysis of a manually annotated database of human drug metabolism information from the Thomson Reuters, including more than 10,000 xenobiotics reactions and more than 1,500 enzyme substrates and 1,000 enzyme inhibitors with kinetic data. So in the Metacore, actually, it is using the similarity search, and in the similarity search, it is using the Tanimoto coefficient. So let's say that here we have two molecules, molecule 1 and molecule 2. How can we calculate the similarity between them? So what is the percentage of the similarity? How similar to this 1 to 2? So when we check this molecule, we have an aromatic ring here, we have an aromatic ring here, we have a carboxylic acid group here, we have a carboxylic acid group here, and here instead of hydroxyl group, we have an oxygen, and then we have a carbon group and a methyl group there. So it looks very similar, but in percentage, how we can calculate? Well, here we need to go to fingerprints. So this is for the first molecule, and this is for the second molecule. So it is checking. I mean, it is making it fragmentize the molecules and checking if these fragments exist in this database. And if it is exist, then we put 1, and if it's not exist, we are putting 0 for the first molecule and for the second molecule. And then we have a Tanimoto coefficient formula. The number of bits set in both is called A. Number of bits set in only 1, but not in the 2, we call B. Number of bits set in 2, but not in the 1. This is called C. And the formula is A divided by A plus B plus C. And if you do this formulation, you will get 0.6. So this means that these molecules and these molecules, based on Tanimoto coefficient, is 60% similar to each other. So we can also do this way. So let's say we have this molecule and this molecule, how similar to each other. So this is the Tanimoto coefficient, as I said. Here we can check number of features, 1, 2, 3, 4, 5. And here 1, 2, 3, 4, 5. So here we have both 5 features. The formula is NAB. So this means that the number of features common to A and B. So this feature, this feature, this feature, and this feature is common. So this means that we have 4 features common, right? Divide by the features in the A, which is 5, plus features in the B, which is 5, minus the common feature, which is 4. And if you do this, you will get 0.67. So this means 67% of these 2 molecules are similar to each other. Okay, is this clear? Okay, in the Metacore we have another. We will get QASR model results, the prediction of the QASR models, but we will also get the Tanimoto Priorizitation value in parentheses. So this value is a maximal Tanimoto coefficient calculated for all molecules in a training set of QASR model. So this means that it will be, let's say, if you have a molecule in your, let's say this is your molecule, then this molecule will be checked the Tanimoto coefficient in the training set one by one. So it will be checked the similarity search by Tanimoto coefficient, and then it will be calculated. It will be given by the highest maximum Tanimoto coefficient value compared to your molecule with the molecules in the data set. And this is called Tanimoto Priorizitation. So this means that the value that is given tells you that your molecule will have maximum similarity of that amount. So let's say if the Tanimoto Priorizitation is 0.70, so this means that among the training set, your molecule is maximum 70% similar to the training set. Okay. So we have already mentioned this. How the binary QASR models predict the therapeutic activity? Well, the prediction of the particular property, so let's say cancer QASR, like the therapeutic activity or toxic effect, is based on the chemistry ability to correlate structural descriptors to that property using recursive partition algorithm. So here you have your molecule with the SMILES code. You will get the fingerprints, and then you will create 3 structures. And based on the experimental and chemical or biological data, it can be physical chemical properties. It can be tried to fit. Okay, so to that end, the training set. So as we said, we have the training set and we have the test set, which was used for the model building. They include chemicals that possess the property or positives and chemicals that do not touch property. I mean, ones and zeros. So there was one question: how these QASR models are actually constructed? What is the target, and so on. So for a prediction of a particular therapeutic activity, let's say cancer, marketed drugs were used if their number was greater than 100. For the cancer, if we have more than 100 marketed drugs, FDA-approved drugs, or drugs in clinical investigation, and if this number is more than 100, then we are using this only. Else, drug candidates in clinical trials and preclinical compounds that's with in vivo activity. I mean, we are not using in vitro, but we are using in vivo activity, have been added to training set. So let's say if you have 50 FDA-approved drugs, in that case, you can use these 50 FDA-approved drugs, and then another 50 can be added from the in vivo activity. For the prediction of the toxic effect, chemicals and drugs that had been annotated in Metadrug database cause a particular effect were used. And as I said, we are using 1 and zeros. The 1, if you get the 1, this means that this is the maximum activity, and if you get 0, this means your molecule is absolutely not active. So these are the parameters that we use for the chemistry parameters. So the default values are path length is 5, maximum segment is 3, p-value is threshold, Bonferroni is 0.99, and so on. So we have these parameters. So for better understand the statistical results, I would like to mention this: an instance where a biometric system identifies users that are authorized and allows them to access is called each of the following. So let's say you have a biometric system that identifies the authorized person, which allows to access to the building. is it a false negative, or is it true negative, false positive, or true positive? So an instance where a biometric system identifies users that are authorized and allows them to access to, so which one is correct? Of course, true positive, right? Because what we are expecting authorized person, which is true, and it is allowing to pass, which is positive. Positive is identified, negative is rejected. Therefore, true positive is correctly identified, and false positive is incorrectly identified, right? So false positive means, so let's say if an unauthorized person, the system allows to enter the building, in that case, it is false positive. True negative, correctly rejected. So let's say that if an unauthorized person try to enter the building and the system does not allow this, then this is called true positive, true negative. And we have false negative, incorrectly rejected. So if I am the authorized person and if I want to enter the building and if the system does not allow me, this is called false negative. Okay. So in the QASR models, we are getting this kind of sensitivity, specificity, accuracy, and multi-correlation coefficient. And the formula of each of them is like this. For example, for the sensitivity, true positive divided by true positive plus false negative. Specificity is true negative divided by true negative plus false positive, and so on. So if you know their meaning, then you can understand easily. Yes, I think I already mentioned all these kind of things. So for example, if I give this molecule, I can get this kind of metabolites, okay? So for example, as you can see here, we have, this is a full structure, but yeah, this is the display structure. And here, as you can see, with the metabolites, it is actually adding this part, right? Yeah, it is like a cysteine transfer. And here also, we have another one. So this kind of molecules can be predicted. I'm not sure if our molecules calculation is already completed, so I can also check. Yeah, it looks it's completed. Yeah, it's done. So if you go here, server is in the UK. It's taking some time to connect. So these are molecule metabolites. So if you want to export metabolites, you should just click here. You can get STF files or Excel files. Yeah. So these are molecules, and these are the metabolites. Yeah, these are the metabolites that is created. So by this metabolites, you can actually get an idea about, if these metabolites still show some activity. So if I will work with this molecule, and if I get these small metabolites, and if I made the docking with the original molecules, then maybe I need to go and check the docking with these metabolites as well, because I know that this, the small molecule will give us these metabolites, and if these metabolites will have this similar docking score with the original molecule, then there will be no problem. But if the docking scores goes smaller values, in that case, we have a big problem. Okay. Yeah, this is, I showed you. And then you can go to models, properties. Here you can actually see the molecular formula, molecular weight, reactive, Lipinski. Yeah, this is, it says that Lipinski rule of 5 is okay. Do you remember what was the Lipinski's rule of 5? Who can tell? Yeah, it's written here. Right? So we have 5. So the molecular weight should be less than 500 Dalton, or 500 gram per mole. The total number of hydrogen bond acceptors should not be more than 10, and total number of hydrogen bond donors should not be more than 5, and the log p value should be under 5. So this is the main rules of the 5 of the Lipinski's rule of 5, and this molecule that we gave, it actually fits this rule. Going to the therapeutic activity predictions, so this molecule that I just wrote has 0.58. So this means that it can have potential to use as an anti-allergic molecule. So the values in the under the therapeutic activity prediction is called TP or Tanimoto Priorizitation, as I described you. So this means that this model has 258 training set molecules and 47 test set molecules. The molecule is actually when you compare with the similarity or Tanimoto coefficient of your molecule with these molecules in the data set, has maximum value of 49%. It's not potential for the Alzheimer, it's not potential for angina, it's not potential for arthritis, and so on. So you can get this kind of green and red colors. Red shows not potential, and green shows it may have some potential. Okay. And also you can get similar compounds. For example, the molecule that I draw has already in the system. You can also get the possible targets. So which let's say which protein targets can be used for this. For example, one of the potential targets is carbon anhydrase 4 or EGFR. So, and you can also click here, and you can go to this network. So it's now building the network, and then you can see which protein targets are actually connected to each other, and which proteins that you can use as a target structure. Here you can get so which I think there's also visualization option. Yeah, you can export image. So from this you can understand which protein targets actually connecting to each other, and they are actually getting the information from different targets. With this analysis, let's say that you actually received some of the molecules. In that case, what you can do, you can let's assume that you screened 500 molecules with the Metacore and against the cancer therapeutic activity, and let's assume that you get the 50 molecules with more than 0.5. You can just focus on these molecules and go to this network analysis and check which protein structures can be considered as a target protein, and then you can use this target in the docking simulations, and you can get the docking scores, and you can check the you can compare the docking score of this target protein with a well-known inhibitors of this target protein. So I would like to show one example that we have already did this to use the Metacore. Yeah, for example, here we screened more than 5,000 molecules to predict the therapeutic activity, and you can see from 1 to 0, and then you can also check the therapeutic activity value with it versus the TP, and then you can get which target structures can be used, and you can do docking, and you can get an idea. So I would like to give a couple of examples that our laboratory has used Metacore Metadrug to identify small molecules against different problems. First algorithm that I would like to show you is kind of fragment-based structure pharmacophore modeling study. So what we have done in this case is we used around 2 million clean zinc fragment-based library, which has molecular weight of 150 to 150 Dalton, and Schrödinger's medical fragment library, which is 667 molecules, which is 2 million molecules. And then we conducted docking with high throughput visual screening, then top 20% with the high throughput visual screening compounds on the Glide SP, and then top 20% with the Glide extra precision. So SP is standard precision and XP is extra precision. So we received 400,000 molecules in after HTVS, 80,000 after SP, and then finally we used 10 heat molecules. So then we used this molecule to generate. Yeah, we used this. Also, we used top 85,000 molecules docking poses from Glide XP to create the ePharmacophore model generation. And based on these ePharmacophore units or hypotheses, we constructed the model and we screened 260,000 molecules from Ottawa, drug-like molecules. Another method that we used was the text mining. So we know that indole is quite crucial elements or fragments in different cancer or anti-cancer drugs, such as the vincristine, which is still using, by the way, vinblastine or mineralbine or vindestine. You will always find this indole fingerprint. So then you can estimate if you want to get a new anti-cancer molecules, can we use in our molecules indole? So what we did, we used SPECS database, which is more than 200,000 small molecules. And then we converted this to more than 200,000 molecules from SPECS to IPAC text file. Now we have in each line, we have the IPAC name of each molecule. So then we selected the molecule indole. So if the indole phrase is actually passing on this line, we kept this molecule. If it is not passing, then we ignored. Then we converted these molecules that has the indole in their phrase to again to 2D structure. And then for these molecules, we conducted Metacore Metadrug binary QASR analysis. So we tested cancer and toxicity. And then based on this cancer and the toxicity values, we selected the molecules and then we used this molecule in the docking. And we selected the molecule for the log-M simulations, and we run the MMGBSA simulation. So here we went two different ways. One of them is selection of the heat molecules and running the directly long-M simulations, 100 nanosecond simulations. Another way is run the short-M simulation, which is 1 nanosecond for all of the molecules, and then get the GBSA analysis and then select from this. So one of them is directly goes to long-M simulation with very small number of molecules, but here we used very large number of molecules for the short-M simulations, and then we calculate the GBSA and then we select the molecules.
Which program did you use for running the molecular dynamics?
Yeah, for the molecular dynamics simulation, we used Desmond here. Desmond. So this is the 5-sided receptor activity, receptor cavity ePharmacophore model. So this is the binding pocket. And in this binding pocket, when we run this fragment library, I mean, there's 200 million molecules in this binding pocket. We received 5-sided AMRDD model. So A is hydrogen bond acceptor. So this one is acceptor. This one is ring. This one is neutral. This one is sorry, negative. This one is donor, and this one is donor. So it is like this. I'm not sure if this will work. Yeah, we get this model, and then we have millions of molecules here, and then we are just trying to fit these molecules on this model. I mean, we are trying to fit the structures and their fragments. And based on this fitness, we are getting the fitness score. So here you can see the docking score and fitness scores. And based on the docking score together with the fitness score, high fitness score, good docking score, molecules is actually selected only. Yes, then we calculate the, we apply for the Metacore Metadrug. And here you can see for the selected molecules, we put them to the Metacore Metadrug analysis and receive the therapeutic activity predictions. You can see here which diseases actually becomes becomes available for these molecules. For example, they are very active, potentially active for depression, heart failure, viral, and so on. Toxicity profiles. So toxicity profiles is, we don't have too much toxicity, a few toxicities here. So these are the pharmacokinetic predictions from the, again, Metacore. We checked if they are passing the blood-brain barrier, if they stay in the, human serum albumin and so on. Then we received, we used a set of molecule in the longer MD simulations. And we checked their protein ligand contact analysis. And we checked, of course, the GBSA and compared with the control ligand. Another example, we try to find TNF-alpha inhibitors. So it's a trimer. So initially we construct the QASR model with the 22 known TNF-alpha inhibitors with their corresponding activities. This is the model that we received. Then we checked the statistical results, which was looking good. And then we used this ligand-based pharmacokinetics model with exon test set, and our model showed good predict, it can predict well the unknown compounds activities. Then we used this model to screen very large number of molecules. So here again, we have the AADDR model. It's something like that. And then after the ligand-based pharmacokinetics model, we selected 400 small molecules and we docked them. And we run MD simulations. In the MD simulations, what we were trying to understand, how these inhibitors that we identify can break the trimers. So here, as I said, it is the 3 units. And we know that this TNF-alpha enzyme becomes active when its structure is actually becomes compact. So if you disrupt the structure, then enzyme becomes inactive. So the inhibitors work like this. So we try to see if these inhibitors will be able to disrupt the conformation. So here, what we did. So during the simulation, we checked the positions of the, the volume of the binding pocket, and try to see if this volume is increasing or decreasing. If it is increasing, this is a good sign that the protein or trimer becomes disrupted. This is another one. This is a known molecule, catechin, and this is our molecule. And then we checked the volume distribution. So how the molecules, our identified molecule, and the known molecule affects the binding cavity size. This is the binding cavity. Yes, here we can see the RMSD, and also we check the volume. Volume is like this. This is the solvent accessible surface area of positive control, and this is our hit molecule. As you can see, when we put our hit molecule, then area becomes larger. So this means that it is actually disrupted disrupted the binding cavity. There's lots of more work, but I don't want to bore you too much. If you have any question. Which one? Metacore?
Yeah.
Unfortunately, it is under license. But yeah, John Bay is here. You can ask the free evaluation. Yeah, except it is the last workshop. If you have any question related with the first two also, I mean workshop 1, 2, 3.
Can you set the presentation on maybe the article itself?
Sure. I can share with you. We have your email, so we can share this presentation to you. I mean, we have one presentation from the Selma that she explained the neural network analysis and construction of the ligand-based models. And then I think we don't have the presentation for the second workshop, but we already shared with you the related files. Yeah, you can actually try yourself all this workshop to see if you are actually will be able to repeat all this stuff, and if you face any problem, you can. Our lab is just one floor below, so we are always here. So you can just come to us and ask your question if you are in Istanbul. You can create yourself the QASR model. Yeah. We are doing this. I mean, so this is the available QASR models, but of course it is quite easy to construct yourself. I mean, Metacore is also allows you, so you can construct your own QASR models if you have enough data, of course. So all of these models mainly constructed based on the known FDA. So let's say if it is cancer, it is not just one type of cancer. Okay, so it is taking so if this small molecule used as an approved drugs against the cancer. Okay, so this is one information, and then you have another one and so on. So this is like if this molecule, if this, let's say, fingerprint in your small molecule is absent or present in the databases. So based on this, it's actually giving you data. So it is not actually. If you want to go with the target, I showed you. You can go with the this network analysis, and you can get which target is possible for this disease. So, for example, if you are using, if you are interested with the pancreatic cancer, in that case, initially you can go with the cancer in general. In that case, you can get a set of molecules, and then you can use these molecules on your target structure related with the pancreatic cancer. So I think we will be all together tomorrow and until Saturday evening. Thank you for being here today. We are very happy to share our knowledge, and we want to continue these workshops during 2024. I think we will have another workshop series, not just on the 10th BAU Drug Design Congress, but we will do a congress, or let's say the workshop, most likely in the February or March, under Biophysical Society meetings. So I just appointed as an ambassador of Biophysical Society BPS ambassador of Turkey. So that I need to organize a few workshops for the undergrad and graduate level students. So that's why I'm planning to make such kind of organization next February or next March. So if you're interested in this, of course we will inform you, so you can also attend this events again. Thank you.